Welcome everyone to our channel. Today, we are comparing how well inactivated vaccines and viral vector vaccines work, and we'll also explain why viral vector vaccines can sometimes lead to vaccine-induced immune thrombotic thrombocytopenia in COVID-19 vaccines. Before we delve into the topic, let's grasp some basic information about the COVID-19 virus. Coronaviruses are a family of enveloped positive single-stranded RNA viruses. 4 types of glycoproteins found in the viral body, and they are the main structural proteins of this virus. They are spike glycoprotein, tiny envelope glycoprotein, membrane glycoprotein, and nucleocapsid glycoprotein. The M protein helps the virus bend and become spherical, as well as maintain the virus basic structure. The viral assembly and release are aided by the protein. SARS-CoV-2 uses its spike protein, SP, an envelope glycoprotein, to enter human cells via angiotensin-converting enzyme to receptor. First, we see the inactivated vaccines. CoronaVac, Sinovac, Sinopharm, BBIB-Picorv, and Covaxin are the examples for inactivated vaccines. In an inactivated vaccination, the virulence properties of live viruses are reduced by heat, UV rays, and chemical treatment, formaldehyde or propiolactone. The pathogen in this vaccine remains in a dormant state. Although this deadly virus cannot cause disease, it can trigger an immune response in the event of a future infection. The inactivation form of the organism creates a secure vaccine, especially for immunocompromised individuals. Moreover, these COVID-19 vaccines showed a lower immune response as compared to the live vaccines, and they also may need many booster doses. In addition, in the host body, the vaccine is unable to initiate a replicating process. It is less dangerous than a live attenuated vaccination and other forms of vaccines. Next, we going to see viral vector vaccines. Covishield, Janssen are examples for viral vector vaccines. Covishield is a monovalent vaccine composed of a single recombinant, replication-deficient chimpanzee adenovirus, ChAdOx1, vector encoding the S-glycoprotein of SARS-CoV-2. Viral vector-based vaccines differ from most conventional vaccines in that they don't actually contain antigens, but rather use the body's own cells to produce them. By infecting cells and instructing them to make large amounts of antigen, which then trigger an immune response, the vaccine mimics what happens during natural infection with certain pathogens, especially viruses. Vaccine-induced immune thrombotic thrombocytopenia, VITT. VITT, also known as thrombosis with thrombocytopenia syndrome, is a life-threatening reaction to COVID-19 vaccines induced via an immune response to the endogenous protein, platelet factor 4, PF4. Mechanism of pathogenesis. 1. Vaccine administration. COVID-19 vaccines that use a harmless virus to deliver genetic material, such as viral vector vaccines like Covishield. 2. Protein binding. This vaccine has parts that stick to a protein on your blood cells called PF4. 3. Immune response. Your body sees this as something new and strange, an antigen, so it makes defense cells, autoantibodies, to fight against it. 4. Platelet activation. These defense cells attach to the PF4 vaccine parts, which causes your blood cells, platelets, to activate. 5. Blood clot formation. When platelets are activated, they can stick together and form blood clots. This can lower the number of platelets in your blood, called thrombocytopenia, and cause blockages in your blood vessels, called as thrombosis. The exact mechanism of VITT is not well defined, and it has not been resolved why cases of VITT are predominated by vaccination with AV vaccines. Most people who get the vaccine will not experience VITT. If you have any concerns about the vaccine, it's best to talk to a healthcare provider. They can give you advice based on the latest scientific evidence and your personal health situation. Thank you, friends. I hope that this session was helpful. Created by MedC Frontiers.